My name is James Kokender from the NCI. I'm presenting a multi-center trial of anti-BCMA CAR T-cell product known as BB2121. The trial is sponsored by Celgene, and up front I'd like to thank my co-investigators in this multi-center trial who are all listed here. First of all, BCMA, a promising target for multiple myeloma. Clearly, multiple myeloma is a disease that is essentially incurable, and new treatments are needed desperately. BCMA is a member of the TNF superfamily. It's expressed universally on multiple myeloma cells, and critically, expression is restricted to plasma cells and only a very small subset of B cells. Most B cells do not express BCMA. We've shown in a phase 1 study at the NCI initial proof of principle for the anti-BCMA CAR T-cell. This was published in Blood in 2016, Ali et al., and there's an abstract today at this meeting. Brudno et al., it's actually abstract 524, and will be presented this afternoon. T-cells transduced on these studies were transduced with a gamma retroviral vector encoding an anti-BCMA CAR with a CD28 costimulatory domain, and significant cytokine release occurred on these studies. The trial that I'm talking about now is with our multi-center trial. The T-cell product called BB2121 utilizes a different CAR. This CAR is encoded by a lentiviral vector and contains the same single-chain FV, which is right here called 11D5-3. It contains a 4-1BB costimulatory domain and a CD3 zeta T-cell activation domain. We showed in preclinical testing this CAR was highly effective in vitro with low tonic signaling and was able to eradicate solid tumors of multiple myeloma in mice. Our protocol design started out with low doses and escalated up in a 3+3 manner. We started with a dose of 50 million total flat dose CAR T-cells, CAR positive T-cells. Then we went to 150 million, 450 million, then 800 million. The general protocol schema was for leukapheresis, and then manufacturing, which took 10 days plus the release testing. Chemotherapy was given as a conditioning regimen to enhance the activity of the CAR T-cells. Fludarabine 30 milligrams per meter squared, cyclophosphamide 30 milligrams per meter squared. CAR T-cells were infused on day 0. Follow-up was conducted with blood draws and bone marrow biopsies. First response assessment was done at week 4. The data which I'll present in detail today is about our dose escalation part of the trial. These are the data that we have prepared and certified for presentation. 24 patients. Clinical deterioration occurred in 3, which led to us only dosing 21 of the patients. And we have 21 of those patients, all 21 evaluable for response. Manufacturing success rate was 100%. Since these patients were treated and follow-up was completed to a certain degree, we've treated 12 more patients on an expansion cohort. I will not present these 12 in detail today because the data has not been fully analyzed yet. The baseline demographics of our first 21 patients. To date, safety profile has been manageable with doses as high as 800 million CAR T cells. 2 reported events of grade 3 CRS resolved within 24 hours. So I've talked mainly about our dose escalation because we blocked our data on that, but I do need to mention one patient in our dose expansion phase who had a delayed onset reversible grade 4 neurological toxicity. So again, this is on the dose expansion phase, and we're not fully reporting the dose expansion phase, but in the interest of full disclosure, we are going to disclose this one grade 4 event which occurred in our dose expansion phase, grade 4 neurologic toxicity associated with tumor lysis syndrome and CRS. Patient recovered completely, the neurologic toxicity resolved. The patient had the highest disease burden on the trial, and the patient has now obtained a very good PR despite low BCMA expression on their myeloma cells. Finally, we have a trial that we're starting up, a global pivotal trial called KARMA, which is open to enrollment. and the BB21 dose range will be 150 to 300 million CAR T cells. Thank you.